अभी रखा है एक मिनट जरा।
अच्छा ये नीचे वो वैसे करना है
खाली स्टार्ट कर दीजिए बस।
Good afternoon to all of you. The second lecture for today is Mycoplasma and Ureaplasma infections. Now we'll be discussing mainly the genital mycoplasmas. You've already had a lecture on gonococcus, that is one of the etiological agents of STIs. We are now continuing with the list, which also comprises of the mycoplasmas and ureaplasmas, which are also etiological agents of STIs. I'll be covering my lecture. This is just the flow of it. In an introduction, the taxonomy of mycoplasmas, which are the genital mycoplasmas, the characteristic features of the main pathological mycoplasmas, what are the infections caused by them, the laboratory diagnosis of genital mycoplasmas, the antimicrobial susceptibility and the treatment of these infections, followed by the concluding remarks. Now, mycoplasmas are the smallest self-replicating free-living prokaryotes which lack a cell wall. That is, like they are bacteria because they belong to prokaryotes, but they are the smallest self-replicating free-living organisms which lack a cell wall. This is in stark contrast to bacteria which have a cell wall. Now they are fastidious because of a very small genome size and a very low GNC content. In fact, initially because of this small size, their small genome, and since they were filterable, initially they were thought to be viruses. But then they were placed amongst bacteria because, like bacteria, they contain both DNA and RNA. They are also susceptible to antibiotics, unlike bacteria, and they are the smallest self-replicating free-living organisms. They can grow on cell-free media. Unlike viruses, in which we need tissue culture systems, they cannot grow in our inanimate culture media, the biochemical synthetic media. Now, the mycoplasmas, they not only infect humans, they also infect plants and animals. Now, amongst the human mycoplasmas, we have around 16 species which affect humans, both the genital tracts and respiratory tract. Today we will be only basically talking about the human genital mycoplasmas, not the respiratory one, but the genital. Now, mycoplasmas belong to the family Mycoplasmataceae, class Mollicutus, order Mycoplasmatales. Now, here we'll just confine to the order Mycoplasmatales, in which we have the human mycoplasmas, that is the genera Mycoplasma and Ureaplasma. The others, as you can see, the Entomoplasmataceae, E. coli plasmataceae, Anaeroplasmataceae, Spiroplasmataceae. These are mainly found in animals and plants. Now, if we think the genital mycoplasmas, the ones which have a proven pathogenic role in man, are Ureaplasma, Mycoplasma hominis, and Mycoplasma genitalium. Now, why as such, you also must remember that mycoplasmas, in addition to being also causing disease in animals and plants, in humans, mycoplasmas are also found as commensals in the genital tract as well as in the respiratory tract. But the pathogenic ones are, which are just enumerated amongst the genital mycoplasmas, and amongst the respiratory mycoplasmas, the pathogenic is Mycoplasma pneumoniae, which is not going to be discussed in this lecture. Now, mycoplasmas have earned the title of being called the second generation STDs, and they are gradually replacing the classical sexually transmitted diseases like gonorrhea and syphilis. Now, the genital mycoplasmas, the three which I've just mentioned, have emerged as important pathogens of the genital tract of both men and women. Now, coming to the first, that is Ureaplasma. Now, Ureaplasma was, in fact, we use the genus as Ureaplasma. We have both Urealyticum and Parvum, the pyovirus. Now, this was first discovered in 1954 as a pathogen causing non-gonococcal urethritis in men. Now, you must remember, you've just had a class on gonococcus. You know what urethritis is. And the leading etiological agent of urethritis is Neisseria gonorrhoeae, followed by, in the cases where gonococcus cannot be isolated, those cases are known as non-gonococcal cases of urethritis. And then the first organism you must think of in your list is Chlamydia trachomatis, followed by the mycoplasmas. So in fact, we have a term now, not just NGU, but NGCU, which means non-gonococcal, non-chlamydial urethritis. So if you have a case of urethritis in which you excluded out gonococcal infection and Chlamydia trachomatis infection, then you think of the mycoplasmas. Now there are two, as I said, you have the genus Ureaplasma, but you have two species in this. Generally, we just say Ureaplasma urealyticum, but actually you have two, Ureaplasma parvum, which is also known as biovar 1, and Ureaplasma urealyticum, which is known as biovar 2. Now, why all these biovars and all came into the picture? Because as I said, Ureaplasma and Mycoplasma hominis both can be found as commensals also. So then, why is it that are there certain strains which are more pathogenic and cause disease in man? So for that, they were divided into the two biovars with further serovars. And there are some studies which implicate Urealyticum as more pathogenic, some with Parvum. Now, the most prevalent potentially pathogenic genital mycoplasmas, which are isolated from the urogenital tract of both men and women. Now, coming to the microbiology of mycoplasmas, as such, they are derived, as I said, from they are prokaryotes, smallest free living, they have evolved from Clostridia-like organisms, but they lack a cell wall. Now, what is the implication of the mycoplasmas not having a cell wall? As you know, that cell wall is the site of action of a very huge group of drugs, that is the beta-lactam drugs. So hence, since these mycoplasmas lack a cell wall, hence they are not susceptible to penicillin and the beta-lactams, which in fact is a major chunk of antibiotics which are used for treatment of many infections. Now they have a tri-layered membrane and they have tip organelles for attachment to the host cells, and since they have a very low GNC content and the concept of a smallest genome, they are fastidious and they require complex nutritional requirements. Hence, these mycoplasmas do not grow on the routine culture media that you have been observing, things like blood agar, McConkey agar. We need a very complex media which we prepare, which is known as the PPLO broth, which is liquid, and PPLO agar, the solid counterpart of it. Now, these are some of the pictures of the mycoplasmas here. Now, a very characteristic growth feature of mycoplasmas is they form on agar fried egg colonies. That is, you have a dense nuclear core structure and a periphery. Now, they have, in case of Ureaplasma, as the name indicates, they have a positive urease test. They utilize urea by production of the urease enzyme. Now, in liquid media, a very characteristic feature of mycoplasmas is that the broth culture, normally when you think of growth in liquid media, you come to know that there is growth in liquid culture media if the broth turns turbid or you have the form of a pellicle or some deposit. Here, the broth remains clear when there is growth of these mycoplasmas. In fact, when there is turbidity, it indicates that there has been contamination by bacteria. Why is it that turbidity is not reduced, unlike bacteria, when mycoplasmas grow in liquid media? That is because of their very small cellular mass. Hence, when liquid broth shows growth of mycoplasmas, it is not will not show turbidity. And these organisms are rarely discernible in a gram stain preparation. So we have to use other stains like Giemsa and the Dean stain for visualization. Now these are just to show you pictures of the colonies of Ureaplasma and their growth in liquid broth. As you can see the colonies, this is the stain that has been used is Dean stain, the Dean stain, and the growth, the media is the PPLO agar, which stands for pleuropneumonia-like organism. You can see on the agar how these are the so-called fried egg colonies. In fact, Ureaplasma, because they produce very small colonies as compared to Hominus, it was also known as the T mycoplasmas, the tiny mycoplasmas. So you can see the dense core and around it the periphery, just like a fried egg. Now this is the media, the growth in liquid media, that is PPLO broth. And here, as I have told you earlier, that the media, as you can see, the liquid media, you do not see any turbidity. Unlike if there was bacterial growth, it would have turned turbid. Now in mycoplasmas, the indication of growth is by exploiting their metabolic properties. So like in PPLO broth, if you have to culture Ureaplasma, then the broth is supplemented. It's the basal broth which contains yeast extract and a very important thing, that is horse serum. Horse serum is required because it requires cholesterol for its growth. And in addition to it, we have to add antibiotics like penicillin to prevent bacterial contamination. Now here, this is a growth of Ureaplasma urinaticum, and as you can see the media, it has the one which is culture positive for Ureaplasma has turned pink because we've supplemented the basal media which contains the PPLO, the yeast extract, the PPLO broth, the antibiotic, horse serum with urea. So if Ureaplasma urinaticum is there, it will utilize the urea by production of the urease enzyme, and we have a pH indicator in red, as a result of which the media, because of the breakdown of urea into ammonia, the pH turns alkaline and the media turns pink, pinkish red, whereas culture, the negative remains yellow straw colored media. So these two pictures show you the growth of Ureaplasma on solid media, that is PPLO agar, where you can see the fried egg colonies, Dean stained, and the liquid media where in the PPLO broth, where you can observe the color change into pink, reddish pink. Now coming to the third and important genital mycoplasma, Mycoplasma genitalium, which in the recent years only has gained a lot of importance. Now this is again, it is in fact amongst the mycoplasmas the smallest self-replicating organism, and it's just 580 kb, coding for fewer than 500 genes, closest to the concept of a minimum cell. This is extremely difficult to culture, unlike Mycoplasma, hominis and Ureaplasma, which I've just mentioned how you can grow them in liquid and solid media. Here, Mycoplasma genitalium is very difficult to culture. Hence, for detection of Mycoplasma genitalium, we rely on molecular methods, mainly PCR. Now, just to tell you a little about the history of the discovery of M. genitalium. Now, as you can see, it was first discovered in the year 1981 when the article was published in Lancet. This is by Dr. Tully and Taylor Robinson. They're like the pioneers of mycoplasmology. Now, as the title indicates, a new a newly discovered mycoplasma in the human urogenital tract. And simultaneously, another article, again by Dr. Taylor Robinson in 1981, Urogenital Mycoplasma Infections of Man, review with observations on a recently discovered mycoplasma. So, M. genitalium was discovered in 1981. Now, this is the transmission electron micrograph of M. genitalium. You can see it has a characteristic flask shape and the terminal truncated portion with extracellular projections. And this basically the flask shaped structure, the terminal truncated portion, that is the one by means of which it adheres to the host cells, the mucosal epithelial cells of the genital tract. Now coming to the characteristic features of M. genitalium, again the same small genome size, GMC content of 32%. Now here it metabolizes glucose, not arginine or urea. Ureaplasma metabolizes urea. Mycoplasma hominis metabolizes arginine. Mycoplasma genitalium metabolizes glucose. Now, this has very slow growth, but it is much more rapid growth upon repeated subculturing. Production of, yes, that is if one tries to grow as such, it is very difficult, but similar colonies like the other mycoplasmas. It has a bottle shaped, as I showed you the transmission electron picture, a bottle shape with a terminal rod-like structure. Here, as mentioned earlier, Mycoplasma genitalium, because of its difficulty to grow, we basically rely on diagnosis by using of molecular techniques. Now this adheres to mainly the attachment protein by means of which it attaches to various human and animal cells, the adhesins. This is the MGPA, that is Mycoplasma genitalium adhesin protein, which is a 140 kDa. There are several other proteins, but this is one major protein which I mentioned because the PCR which we do for detection of M. genitalium is the one for which the gene target is what we use is MGPA 140, the kDa protein which is being targeted. The gene. Invasion of epithelial cells by means of this also, it helps in adherence and also the invasion of the cells. This organism shows gliding motility and it is inhibited just like the other mycoplasma. Since it lacks a cell wall, it will not be susceptible to penicillin and the beta lactams, but it is susceptible to tetracyclines, fluoroquinolones, as well as the macrolides. Now in recent years, Mycoplasma genitalium is gaining a lot of importance because first it is being isolated in large numbers from cases of NGCU, that is non-gonococcal, non-chlamydial urethritis. And the problem is that it is emerging as, in fact, as they say, the next superbug, because there are reports that high incidence of resistance is developing, especially to tetracyclines and macrolides, and now even to the fluoroquinolones, that is moxifloxacin. Now, what are as I've just given you a brief preview of the genital mycoplasmas, that is Ureaplasma, Mycoplasma hominis, and Mycoplasma genitalium. We've talked about a bit of their microbiology, their structure, their growth morphology, colony characters. Now, what are the infections caused by them? Obviously, as the name genital, they cause genital tract infections in both men and women. Now, this table shows us the relationship between these organisms with disease. Now, this is the problem why all these associations had to be developed, because they are, especially for the case of Ureaplasma and N. hominis, they are found as normal commensals of the genital tract also. Now, the first condition is, as I said, non-gonococcal urethritis, for which a strong association they have been shown to cause. It has been associated strongly, that is M. genitalium has been associated strongly with conditions like non-gonococcal urethritis, even including Ureaplasma species, that is both parvum and urelyticum, which have been implicated with non-gonococcal urethritis causing. So it is a definitive cause of non-gonococcal urethritis, both Mycoplasma genitalium and Ureaplasma species. As you can see, Mycoplasma genitalium has also been is a cause of not only acute non-gonococcal urethritis, but also chronic non-gonococcal urethritis, blepharoposthitis, chronic prostatitis, less just a 1 plus, epididymitis, then Rita's disease, that is the sexually reactive arthritis. Now in women, we can see both M. hominis, Ureaplasma, and M. genitalium have been sort of an association with bacterial vaginosis. Then it is a cause, M. genitalium is strongly, it has been a cause of cervicitis and then infertility. Because once infection occurs, there'll be inflammation and then scarring of the tubes, which this is a sequelae of infection of these agents, that is infertility in women. That is why, especially in women, it is important to screen them and to treat before you have serious sequelae of infertility developing in women. Then another thing is, in case of Ureaplasma, it is also, if you can see down in the table, it not only causes genital tract infections in men and women, it is also a very important cause of neonatal respiratory disease. That is, it is chronic lung disease of the newborn. When you seen in premature babies, when the respiratory tracts, if the mother is infected and the respiratory tract gets colonized with this organism at the time of delivery, that is a time then because of the immature immune response of the babies, excessive secretion of these interleukins, it can cause chronic lung disease of the newborn, where the child still requires oxygen support till the age of 28 days. So it's Ureaplasma, the infection is not only confined to the adults, but also to newborns. Now, just to repeat the spectrum in adults, urogenital tract infections, cause of non-gonococcal, non-chlamydial urethritis in 30% cases, pelvic inflammatory disease, salpingitis in women, then the sequelae being infertility, Ureaplasma, especially the cause of adverse pregnancy outcomes like chorioamnionitis, spontaneous abortions, and stillbirth. Then, like most STIs, they are also co-pathogens in HIV-infected individuals. Now, these organisms potentially increase the susceptibility of acquiring and transmitting HIV. Now, another spectrum of infection caused by Ureaplasma, in addition to adults and newborns, prematures, babies, is also they may cause infection stones, that is renal calculi, which are of different types. You have the struvite, you have the metabolic stones. The Ureaplasma causes the infection stones. These are by virtue of its ability to produce urease enzyme. It creates an alkaline pH and it causes these, they are called the staghorn stones, their composition being ammonium magnesium phosphate and calcium phosphate. And they are staghorn type, they can cause severe obstruction. And the importance is that even if you remove them, you have to treat the infection which is the cause of the formation of these stones. Otherwise, you will have recurrence of these stones, the infection stones. Now, neonates, I've already told, the infection caused by Ureaplasma, chronic lung disease of the newborn may rarely cause bacteremia and even CNS involvement like intraventricular hemorrhages, hydrocephalus, developmental delays. Now, just to give you a brief review of the Mycoplasma, what is the status of genital Mycoplasma infections in India? There are very few labs which are working on genital Mycoplasmas in India, so I thought you should know that AIIMS is one place where work is being conducted on genital Mycoplasmas. Here we've had a study in which we did the detection and biomarker discrimination and how co-infections, just this article shows you that we've had co-infections with genital Mycoplasmas and Chlamydia trachomatis infections in patients with genital tract infections at our hospital. As I had mentioned, that most STIs can occur as co-infections and they promote the other STI along. This was another study from India where ureaplasma as a cause of upper urinary tract stones, then ureaplasma as a cause clinical significance diseases in newborns, then ureaplasma, so many co-infections of these agents together with ureaplasma, M. hominis, and chlamydia trachomatis in an HIV positive woman with vaginal discharge. Then co-infections not just confined to the bacterial STIs, but also with the other STIs, the viruses like herpes simplex virus in an HIV patient. Just to highlight that how these STIs can co-infect a person. Then prevalence of these mycoplasmas in treatment-naive HIV infect patients. Just a case study. We had a case of a 29-year-old bisexual male who presented to the STD clinic at AIMS with urogenital discharge. On examination, he had urethral discharge along with genital and anorectal warts. Clinical diagnosis was recurrent HPV, human papillomavirus, with urethritis. And these were just to recapitulate, these are the panel of which was tested, the STI panel, which included both ureaplasma, M. hominis, genitalium, chlamydia trachomatis, and serology for HIV, VDRL for syphilis, TPHA, hepatitis B, hepatitis C. And we found that the patient was culture positive. He was HIV positive, VDRL reactive, that is, had infection with syphilis, Treponema pallidum. He was culture positive for ureaplasma as well as PCR positive for ureaplasma, not only at the urethral but also at the oropharyngeal site. So now you must understand when we come to talk about these STIs, because of aberrant sexual practices, these organisms are now also being isolated from the extragenital site, mainly the rectum. Oropharyngeal, yes, gonococcus is quite often, but these agents are relatively less isolated from the oropharyngeal site, but rectal site, yes. So just to carry home the message that now when we test patients for STIs, one has to take the history, their sexual orientation. In the case of MSMs and all, it is also advised to collect a rectal swab, not essentially oropharyngeal only if he gives a history suggestive that you need an oral pharyngeal swab, but a rectal swab must also be collected, and these organisms are being now isolated from extragenital sites. So here we had a patient who had oropharyngeal and urethral ureaplasma infection co-infected with HIV, Treponema pallidum, and human papillomavirus, to show that how often co-infections with STIs occur in patients. Now, an important thing, as I said a few years before, we were never talking about, we were not aware of these extragenital infections, but now we know that extragenital ureaplasma, mycoplasma infections are occurring, which are a cause of concern, and you need to have awareness about them so that extragenital screening is also done. Now, extragenital infections have been rarely reported. We've reported a case of one patient. Now here next is M. genitalium infections in MSMs. This is another important thing which you must now know about. Be aware of it. We've had a study from our center and we found that in MSMs, the anorectum was the most frequently commonly infected site, followed by the urethra. So it can occur even exclusively. It's not that you need co-infection of both sites. You can have independent rectal infection depending upon the sexual history of the patient. We did not find any oropharyngeal infections, and most of greatest concern is most of the anorectal infections were asymptomatic. So that is why it is important that when you screen a patient coming to the STI clinic, when you are taking his sexual history, if it is suggestive, if he's an MSM, irrespective whether he has complaints at the extragenital sites, rectal swab should be ideally collected because incidence of infection, and if you have infection at the rectal site, the treatment becomes a little more prolonged because it's a little more difficult to treat infections at the extragenital site. Now, laboratory diagnosis, we know it's the type of the specimen, collection, transportation, processing, and identification. Now, non-culture methods, for Mycoplasma, hominis, ureaplasma, we don't have ELISAs and all available. Unlike pneumonia, where commercial kits ELISAs are available, we basically rely upon the PCR. Now, what is the you can culture ureaplasma and mycoplasma hominis, but why do we need these molecular tools? Because we know conventional methods of culture are time consuming. The media is, because they're fastidious organisms, the media is very expensive and it requires a lot of expertise. Hence, PCR is the diagnostic modality. And we know the advantages of PCR, which is true for all agents. Now, the PCR is being done for ureaplasma. It targets the urease gene. And we also have to detect the biovars and serovars. Then we have we have PCR for hominis, which is targeting the 16S rRNA gene. Then we have PCR for M genitalium, which is, as I said, targeting the MGPA, the adhesine gene. Now, after that, so mainly for ureaplasma and mycoplasma, we do both culture and PCR. For M genitalium, we are doing PCR. Now, in addition to that, why initially antimicrobial susceptibility was generally not done for mycoplasmas because they were susceptible mainly to the macrolides and tetracyclines. But now it is advised at times that if the patient, you have reports of patients not responding to treatment because of resistance being reported. So antimicrobial susceptibility is also being done for mycoplasmas because of the reported increase in resistance to antibiotics. Now, this resistance is mediated by mutation and acquisition of new genetic material, and now this has prompted in vitro testing for older and even the newer drugs available. Now, the method is the microbroth dilution method. Some of the antibiotics which are being used for testing is zithro, oflox, josamicin, and doxycycline. Josamicin still is not available here. This is the microbroth dilution test which we do for detection of antibiotic susceptibility, because we really can't do, the colonies, difficult growths, we can't do the normal disk diffusion method which we employ for bacteria. Now, we have limited studies on treatment, and we know the antibiotics are mainly those which inhibit protein synthesis, so they're susceptible to tetracyclines and macrolides, and now, in fact, quinolones are emerging. Now, this is a table which shows the susceptibility. Now, as you can see, ureaplasma is susceptible to both tetracyclines, the macrolides, and the quinolones. M. hominis is inherently resistant to macrolides, that is azithromycin. So that's when the syndromic management, where we are giving treatment of azithromycin, just stat 1 gram. Here, if it is M. hominis, we will have to give doxycycline because it is inherently resistant to macrolides. M. hominis is mainly to quinolones and tetracyclines, and M. genitalium is susceptible to tetracyclines, macrolides, and quinolones. But now we have a lot of resistance, especially being reported in M. genitalium, and resistance to azithromycin. So hence, to conclude, the role of genital mycoplasmas in many human diseases is now established. Because of the difficulty to culture these organisms, this led to an underestimation of their role in clinical entities. The use of molecular diagnostic tools will enhance the diagnostic efficiency, and earnest efforts to diagnose and treat genital mycoplasma infections should be done to prevent the clinical sequelae of undiagnosed and untreated infections. Thank you. These are the two predominant scientists who've done a lot of work in mycoplasmology, Dr. Taylor Robinson and Dr. Jensen.
यहाँ हो गया अभी गए क्या मैडम?